Greetings. Thank you all for joining us today. My name is Matt Hubeck. I'll be the moderator for today's webinar. If during the webinar you have any viewing difficulties, please hit F5 on your keyboard to refresh the webinar window. The audio for today's webinar will be coming from your computer, so please make sure you turn your volume up. Also, at any time during the webinar, feel free to submit questions for our presenters. After the presentation is completed, we will address your questions in a Q&A session. With that, I'll turn things over to Wayne Patton so he can get started. Wayne?
Thank you, Matt. I'm Wayne Patton, Chief Scientific Officer of Enzo Life Sciences. Just as a brief introduction to me, I've been in the fluorescent probe technology area since 1993, where I was joined the faculty of the Photonics Research Center at Boston University. After that, I held positions at Molecular Probes in Eugene, Oregon, and Perkin-Elmer in Boston. I've been Chief Scientific Officer of Enzo for the past 4 years. I'll be handling the dye-based cell assays, whereas Paul Held will be discussing the actual automation of the assays on the biotech platforms. So toxicity testing in the 21st century has changed dramatically from what was previously done. The U.S. National Research Council has a vision for the 21st century to move toxicology from a predominantly observational science at the level of disease-specific models to predominantly predictive science focused upon a broad inclusion of target-specific mechanism-based biological observations. So this includes chemical characterization, toxicity pathways, targeted testing, and dose response and extrapolation monitoring. There's been a paradigm shift in early safety assessment coupled with the drug discovery process. Previously, most toxicology was done late in the drug development pipeline in the preclinical, clinical phase 1 areas. There's been a tremendous emphasis to shift the assessment to the earliest phases of drug development to implement faster, less expensive, and more predictive methods to identify toxic liabilities early in the drug development process. Cell-based phenotypic analysis has become an important tool in this aspect. It's important in the tox area, but it's also important in the secondary screening area. A recent publication in Nature Reviews Drug Discovery points this out very dramatically. Preclinical strategies to identify potential drug candidates can include a variety of strategies, including target-based screening, phenotypic screening, natural substance modification, and biologics-based approaches. An analysis of the U.S. Food and Drug Administration drugs that have been approved by that organization between 1999 and 2008 have demonstrated that the contribution of phenotypic screening to the discovery of first-in-class small molecule drugs has exceeded that of target-based approaches. This approach has been successful in a drug screening capacity, and it's also highly applicable in the toxicology aspects, particularly if you're trying to push the tox investigation earlier into the drug development pipeline. So basically, previously the industry standard has been rather non-predictive, simple in vitro assays of proliferation, viability, and death, coupled with poorly predictive animal testing. This has led to high failure rates, high costs, long development cycles, and high safety liabilities. The approach that Enzo and BioTek are working together on is predictive in vitro cell-based assays that monitor subcellular organelles and pathway functions, including nuclear activities, mitochondria, autophagy, lysosome pathways. Automated high-performance microplate assay workflows are being developed and benchmarked. And our hope is that this will lead to lower failure rates, save tens of millions of dollars in the drug development process, lead to shorter development cycles, and lead to lower safety liabilities. There are a number of instances where drugs have been introduced to the market only to be found to be toxic later on. And one of the biggest areas has been in hepato and cardiotoxicity, where the liabilities based upon the mitochondria were not detected in the initial toxicology analyses and were only discovered later on. So at Enzo Life Sciences, we've been developing celestial kits for cell analysis. These are basically turnkey assays developed to look at various aspects of cellular biology with a particular focus on toxicology-based analysis. The platform is developed to work in the flow cytometry context on fluorescence microscopes or high-content screening instruments, and most important for today's talk, on conventional microplate instruments, and provided they have the capabilities that we're describing today, which is why we actually work with BioTek extensively. Their instrumentation has been developed to allow for smooth workflows in doing the liquid handling and analysis of the cell-based assays. This picture highlights the novel organelle and pathway targeting dyes that we've been developing at Enzo Life Sciences over the past 4 years. We pretty well covered the cell in great detail with multiple probes addressing various subcellular organelles. With respect to toxicology, in vitro toxicology, the nuclear ID chromatin condensation assay is a convenient microplate-based assay for measuring apoptosis. The Ross superoxide assay measures reactive oxygen species. The MitoID membrane potential assay measures mitochondrial energetics, getting back to cardiotoxicity and hepatotoxicity that I mentioned earlier. This is a good assay for identifying and screening out those types of compounds. LysoID red assay measures lysosomal compartment expansion, particularly useful for measuring phospholipidosis. The CytoID autophagy assay measures autophagy induction. This is a process where the cell actually eats itself, digests its organelles and proteins. The proteostat agrosome assay measures aggregated protein accumulation in various structures, and it really depends upon the organ what the name of those structures are. They're called Lewy bodies in the context of liver toxicity. They're called Mallory bodies in the context of liver toxicity, Lewy bodies in the context of a neurological disease such as Parkinson's disease. The other assay I'll highlight quickly is the efflux ID assay, which measures ABC transporter drug activity, and that's key in the resistance of certain cancer cells to drugs is mediated by their ability to pump them out of themselves and avoid the toxic response. So I can't highlight all of these assays today, and I've selected 3 to talk about in detail, and Paul will follow up with automation of those 3 plus 1 additional assay. I'm starting to talk with chromatin condensation assay. And the classical methods for measuring chromatin condensation have been transmission electron microscopy, or the use of a UV light excitable dye called Hoche. The transmission electron microscopy approach is difficult and low throughput. While the use of Hoche is complicated in a number of ways, flow cytometers cannot really, conventional flow cytometers cannot really quantify Hoche, and there are issues with it in the microplate context as well, which I will describe shortly. So what we've done is developed a novel 488 nanometer excitable green emitting DNA probe for looking at chromatin condensation. And this probe allows analysis of chromatin condensation by fluorescent microplate reader, fluorescence microscopy, or flow cytometry. The basic mechanism of action of this dye is that it intercalates within the DNA duplex. The environmentally dependent dye exhibits a large fluorescent signal enhancement upon binding to the DNA. And the signal increase provides a proportional response to quantitation of the DNA. There is, in addition, a further increase in signal upon condensation of DNA during apoptosis. And at the bottom of this slide, I showed the various stages of nuclear condensation, with the last stage being that the chromatin actually condenses into a small ball, and that increases the fluorescence of our nuclear ID chromatin condensation dye by as much as 40-fold. And I'm showing some images of this just to give a feel for the assay. We have control cells and staurosporin-treated cells, both phase contrast and fluorescence microscopy. And in the fluorescent microscope image for staurosporin, you can see the brightening of the nuclei in response to nuclear condensation. On the right of the slide, I'm showing the flow cytometry readout, and a shift in the fluorescence is seen as an increase in fluorescence to the right. So there's a movement of the fluorescent signal to the right as the cells respond to the staurosporin. This slide shows the same assay done in a microplate version using the BioTek instrumentation. And you can see that it's very straightforward to generate a dose-response curve for staurosporin induction of apoptosis in these H-meso cells. You can readily calculate the EC50 value of your apoptosis-inducing agent. And the assay, when it's automated, gives a decent Z-prime value, so you can use it for high-throughput screening activities. Camptothecin is another apoptosis-inducing agent. In the case of Jurkat cells, it really it's not a very potent inducer of apoptosis, and this is shown in the flow cytometry. We have almost no nuclear condensation occurring in this situation. If you use host dye to analyze apoptosis induced by camptothecin, however, you'll get the impression that it is a strong inducer of apoptosis. In the upper left, I'm showing camptothecin-treated cells stained with host, and you see an increase in the signal in the microplate reader as we increase the concentration of camptothecin. On the upper right, though, you see that all of that increase is not due to the dye itself. If we leave the dye out, we still get that increase in signal. That's due to the fact that small molecule compounds tend to fluoresce in the blue region of the spectrum, and that can mislead you into thinking that you have an apoptotic event when you don't. So in the lower left corner, we're doing the same experiment with the nuclear ID green and demonstrating that actually the doses of camptothecin used in this situation did not induce apoptosis in the cells. If we go over to the lower right, you can see in the blue channel, we see the fluorescence of the small molecule compound. So basically, the nuclear ID chromatin condensation kit has an intercalating dye that's with superior permeability for most live cell types. Dye can be used over a wide range of cell densities. It can be used in microplate assay to detect chromatin condensation arising from drug treatment. And one particularly important benchmark is that blue fluorescing small molecule compounds will not interfere with your assay. The next assay I want to profile is our autophagy assay, and we have the world's first fluorescent dye-based autophagy assay. And as I said earlier, autophagy is a process where it's a very regulated process where we have an engulfment of proteins, protein complexes, and organelles into a double membrane sequestering vesicle. And The autophagic cargo is then delivered to the lysosome, where it is degraded into its essential constituents and recycled back to cytoplasm. This autophagy is generally a defense mechanism. It's a way the cell hibernates when exposed to a caustic agent. It lowers its metabolism down to basal levels and tries to survive another day. However, if the process goes for too long, you will have autophagic cell death, and this is one of the forms of programmed cell death in addition to apoptosis. Classical methods for looking at autophagy, double membrane vesicles have been identified by transmission electron microscopy, and various lysosome staining dyes have been used to stain autophagosomes, or more appropriately autophagolysosomes. In addition, non-physiological protein mutations or genetically engineered cell lines have been developed to monitor a protein called LC3 and its association with autophagic vesicles using GFP-tagged protein constructs. Our approach, though, is to use a cationic amphiphilic tracer to highlight the various vacuoles associated with autophagy. The dye has been designed to minimally stain lysosomes through a pH clamping mechanism. There's no requirement for non-physiological protein mutations or genetically engineered cell lines. There's no 18-hour transfection step. It's a very fast stain that occurs in about 10 to 15 minutes. The assay is suitable for fluorescence, confocal microscopy, and high-content screening application. The live cells are stained, facilitating kinetic analysis of the autophagic process, and the assay is compatible with fluorescent microplate readers, allowing easy quantification of the cell response. So the classical method is transmission electron microscopy, and I'm showing an example of that here, where you had to physically score the formation of the double membrane vesicles arising from autophagy. Obviously, that's a very labor-intensive and low-throughput approach to analysis of autophagy. In addition to that, Western blotting has been used to look for the formation of LC32, which is a phosphatidyl ethanolamine tagged LC3 that associates with the autophagosome. And the method on the bottom is looking at redistribution of LC3 using GFP tagged LC3, looking basically for a diffuse distribution and becoming a punctate distribution, and basically counting the punctate spots to determine that there was an increase in autophagy. Again, a rather labor-intensive approach. One dye-based method that's been used is monodansyl cadaverine, and I'm showing control versus starved cells here. And the main problem with that particular dye is that it stains lysosomes as well as the autophagic vacuoles. So you have a very high background staining and are really looking for a very slight increase in fluorescence over this high background staining. The CytoID autophagy detection dye was developed to overcome the limitations of the previous methods. It's a cationic amphiphilic tracer dye that rapidly partitions into the cells in a similar manner as drugs that induce phospholipidosis. Careful selection of the titratable functional groups of the dye prevent its accumulation into the lysosome, enabling labeling of the vacuoles associated with the autophagy pathway. And we've done extensive mapping of this dye with respect to the autophagy pathway. We've done a very detailed pharmacological characterization using our Screenwell autophagy library. And basically the results of that there it is. Okay. The results of that are shown in green. These are the components of the autophagy pathway that we've demonstrated by pharmacological methods are highlighted by the dye. You'll notice that the lysosome in the upper right there is excluded from the green circle. It's not detected by the autophagy dye, and that's what gives us our low background. The red circle indicates lyso ID a dye that I'm not really going to talk about extensively today, but Paul will mention with respect to automation. The lyso ID is focused on the lysosome, the autophagosome, and the autolysosome. So these two dyes highlight different aspects of autophagy. The problem with monodenso-cadaverine as a dye is it detects everything on the page, both the green and the red circles. So it's very difficult to selectively detect the components of the autophagy pathway. So the CytoID autophagy detection kit is the only assay that identifies autophagy regulators in live cells by imaging and flow cytometry. You can see in the microscopy image control cells where we're not staining lysosomes, so we have a pretty empty background. The blue in that picture is Hoche, detecting the nucleus of the cells. And then simply by starving the cells for 1 hour in Earle's balanced medium, we see the formation of these green vacuoles indicating activity of autophagy. In the lower right corner, we're showing the same experiment by flow cytometry where the control cells are peaked with low fluorescence. We starve for an hour. There's a tremendous shift in increase in the fluorescence. And then when we refeed the cells, they return back to the control levels. Here we're using the BioTek automated solution for detecting autophagy in a microplate assay. And we're looking at two different inducers of autophagy, tamoxifen and verapamil. Tamoxifen acting relatively early in the autophagy pathway, and verapamil more in the autolysosome end of the pathway. And you can see that we're able to monitor autophagy. This is a dose response curve, and we can effectively determine what dose of the particular drug will induce autophagy in these cells. So the CytoID autophagy detection kit rapidly quantifies autophagy in native cells. No transfection is required, so it's a very quick stain, taking about 15 minutes. We have detailed protocols on differentiating between autophagic flux and autophagolysosome accumulation. Negligible staining of lysosomes reduces the background. And we've extensively validated the protocol using inhibitors and activators of the autophagy pathway. The last dye that I'm going to talk to before Paul talks about the automation is mitochondrial membrane potential assay. Again, mitochondria are a very sensitive detector of cell toxicity. When the membrane potential decreases, that's an indication that the cell is under assault by the particular drug that you're adding to them. So the classical methods for looking at this have been lipophilic vital dyes like rhodamine 123 tetramethylrhodamine, as well as a dual emission dye called JC1. All of these dyes have had liabilities in the past, and these are dyes I worked at quite extensively and was at Molecular Probes. So we tried to develop a next generation dye that would improve upon the performance of these older dyes. Though JC1 is widely used to monitor mitochondrial membrane potential, it is poorly water soluble, requiring numerous washing steps in a microplate assay. Even at 1 micromolar concentration, the dye tends to precipitate in aqueous solutions, so the analysis is usually confounded by precipitates which lead to high background. The MitoID dye has been engineered to be more water soluble than JC1, facilitating live cell staining without washing or medium removal. Now, we do both protocols. We have done protocols where we do a washing protocol, but we also have done protocols where we just add the dye directly to media containing serum and doing the analysis that way. So the dye basically localizes into the mitochondria, and upon entry into the mitochondria, changes color from green to orange due to aggregation of the dye. The dye forms a specific aggregate called the J aggregate, which has a higher emission wavelength maximum than the unaggregated dye. When the mitochondria When the mitochondrial membrane potential decreases due to a particular drug, the dye leaves the cell and reverts back to the monomeric form, which is green. So this is illustrated in the upper left where I show two mitochondria. The green mitochondria represents the depolarized situation where the dye is in a monomeric state. The red mitochondria is the energized state where the dye has been concentrated into the mitochondria and due to the J aggregates has shifted into this orange-colored fluorophore. You can see that pictorially below the pictures of the mitochondria, and you can see that in the middle image we have orange fluorescence showing active mitochondria. The green fluorescence is cytoplasmic dye, and during de-energization of the mitochondria you lose that orange signal and the green signal actually increases. Here I'm demonstrating that the activity of the MitoID dye relative to JC1. Basically, the MitoID dye is roughly 10 times more sensitive to JC1 in detection of the loss of mitochondrial membrane potential. So when we use a compound like CCCP, we see a loss in the mitochondrial membrane potential with a much lower dose of CCCP than obtained with JC1. This particular experiment was done with washing, and the standard deviation of the measurements are better with MitoID than with JC1, as you can see in the graph, and that's due to the fact that it's very difficult to wash the JC1 out of the cells. The aggregates form on top of the cells, and they're difficult to get rid of. Here I'm demonstrating live kinetic analysis of mitochondrial membrane potential. Here we've taken advantage of the BioTek dual dispenser for the Synergy instrument and are adding the dye directly to cells growing in media. And then we're following the mitochondrial membrane potential as a function of time. The green line shows the control cells where the mitochondrial membrane potential is being maintained. The red is a relatively high dose of the rotenone, and you can see complete loss of the mitochondrial membrane potential. And then the two lines in between, the blue and the purple line, show intermediate doses of rotenone where we are able to capture dynamically the loss of mitochondrial membrane potential in the cells. MitoID membrane potential cytotoxicity kit is 10 times more sensitive than JC1 with superior aqueous solubility. We have a photostable dual emission dye that we're employing in this kit, suitable for time course studies evaluating intact and compromised mitochondria. We have two protocols, a wash protocol and a no wash, no medium removal protocol. And if you're interested in measuring mitochondrial mass, there's a separate dye, a MitoID red, that has been extensively analyzed and benchmarked to measure mitochondrial mass. One application that we use commonly with that is measuring mitophagy or mitochondrial autophagy. So at this point, I'd like to turn the presentation over to Paul Held, who will discuss automation of the celestial assays.
Hello, my name is Paul Held. Thanks, Wayne, for a great introduction. So I'm the laboratory manager at BioTek Instruments for the Applications Lab. I've been with BioTek for almost 18 years. And really what I'm going to talk about today is automation of some of the celestial dyes using one of our instruments, particularly the EL406 washer dispenser. So this washer, as I alluded to, is both a washer and a dispenser. It has a number of different features that enable it to be really a useful tool for the celestial dyes. First of all, like I said, it's a plate washer. It has a dual-action washer manifold that will wash 96 wells simultaneously. In addition to that, it has a peristaltic pump dispenser which basically has 8 tubes that will allow the addition of 8 different reagents if you needed to, but it also will allow for the rapid addition of sterile solutions, and I'll talk about that in a minute. Although in this picture you can't really see it too well, attached to the washer dispenser is also 2 syringe pump dispensers, so it has a total of 3 dispensers in addition to the washer manifold, so that there's a great deal of flexibility in terms of adding reagents. The box that I just indicated here is actually where the dispenser manifolds all kind of meet the plate, if you will. In addition to the washers and dispensers, it also has a priming trough that contains a sonicator, which allows for automated cleaning of the washer manifold, which in many washers is actually kind of an arduous and difficult process. And last but not least, it has a mobile carrier that will accommodate both 96 and 384-roll plates that will basically index the plate to allow for 384-roll plate usage as well as 96. So in regards to the washer manifold, I mentioned that it's a dual-action manifold, and really what this means is that the aspiration and the dispense tubes can be controlled independently, at least in terms of height. This allows for offsets to accommodate smaller wells, such as the 384-roll plates. It allows for different heights for aspiration as well as dispense, so those can act independently of one another. And then with certain options, you can actually get a 1536 well washer as well. Oops, let me back up. One of the key features of the washer in regards to the celestial assays is its ability to wash loosely adherent cells. One of the features that's kind of hidden within the washer is the fact that it contains separate plumbing for very low flow rates, which allows for dispensing of fluids to loosely adherent cells without actually washing them off the plate. As you can see in the upper right, if you wash plates with normal wash speeds and they're loosely adherent, you can certainly blow away many of your cells, which in essence reduces your signal dramatically. Using the same cell types with a different dispense profile that's designed for loosely adherent cells, you can see that after washing that you don't really lose the cells. And it all is accomplished by having basically a separate plumbing path for relatively low slope dispense rates. In addition to the washer, like I said, the EL406 has a peri-pump dispenser, and as you can see on the left-hand picture, that there's it's a peristaltic pump with 8 different tubes. These eight individual tubes can certainly be put into one reagent and one reagent added to all the wells, but under certain situations you can actually use these to add 8 different reagents. Another great feature of the peristaltic pump is the fact that the peristaltic tubing cassette is autoclavable, which makes for easy dispensing of sterile fluids or even dispensing of cells. And as you can see on the right, really what we've done, we've dispensed mesothelioma cells, and I'll talk about those cell type later. But we can certainly dispense those cells and then 24, 48, or 72 hours later still have a sterile, viable solution. So this is an easy means to dispense cells to a plate at the beginning of an experiment. To kind of summarize, one of the things I want to point out is the fact that the 406 is actually small enough to fit into a biosafety cabinet, which it certainly enables for the use of the instrument in situations where you need sterile environment. Besides the washer, like I said, there's 3 different dispensers, so you can really have 3 different reagent additions as well as the wash procedure. The wash buffer itself can actually you can actually have up to 4 different wash buffers on the washer manifold. In addition to that, besides the peri-pump, I didn't really talk about it, this is also syringe pump, so you really have a choice of dispenser technologies available on this one instrument. So what I want to do now is kind of switch gears away from the instrument itself and talk about 4 different dyes that we've automated in regards to at least cytotoxic dyes, cell dyes from Enzo that we've automated. And this automation is actually relative to toxicity assays, and particularly the treatment of mesothelioma. Mesothelioma is a cancer of the mesothelial cells. These cells line the pleural and abdominal cavities. It's a tumor that's been associated with asbestos exposure. It's certainly gotten a great deal of publicity and press, primarily from the legal profession, but in reality it's a relatively rare tumor, and there's less than 5,000 cases per year. But its key element is really it's the only tumor that's been closely associated with an environmental exposure, particularly asbestos. So in regards to mesothelioma treatment, we were interested in that in regards to treatment with a particular agent. And this agent is thiostreptone. It's a naturally occurring cyclic antibiotic. It was discovered in the mid-50s. It has poor water solubility and as such it's primarily historically been used topically. But what has been noted as of late is that there was a great deal of activity for this compound against breast cancer cells, and that as such we were interested in seeing whether or not this agent would also be amenable to using as a treatment for mesothelioma cells. We do know from biochemistry that it inhibits the oncogenic transcription factor forkhead box-M1 or FOXM1, and this was incredibly intriguing to us, primarily because we know that mesothelial tumor cells express FOXM1 to a great deal of degree. It's hard to tell. The staining on the right is a mesothelial tumor tissue slice with a great deal of FOXM1 staining. So that was the rationale for looking for thiostreptone to see whether or not it was a cytotoxic agent to mesothelioma cells. So the first assay I want to talk about is the MITO ID membrane potential cytotoxicity assay. I won't really go into the biochemistry, as Wayne's done a great job explaining that already, but just so you know, it's the MITO ID assay that we're talking about now. In regards to the assay procedure, really what we're using the 406 washer dispenser for is a number of things, and these are outlined in red. The first thing we use is the peri pump to seed the cells. So we can automatically seed all 96 wells of the plate. At that point, we let the cells grow overnight. We then added our drug dilutions manually in this case because we're looking at different drugs. But the nice feature about the washer dispenser is we can then use it to aspirate media and add our dye. So this would be, in essence, a no-wash solution or a one-cycle wash solution. And then follow the incubation from the addition of the reagent, we then read it on one of our BioTek readers. So that's in essence, that's the task. We use the peri pump dispenser as well as the washer manifold on the 406 to really automate the assay. In regards to performance, as you can see on the left, really what we're looking at there is a known agent on H-meso cells. The CCCP certainly reduced orange fluorescence, as you can see in the upper left, and actually in the lower left, you can actually see the green fluorescent increasing in a coordinated fashion with the loss of orange fluorescence. In regards to thiostreptone, as you can, what we're interested in is whether or not thiostreptone actively induced problems with the mitochondria. Unlike CCCP, which definitely caused a decrease in polarization, as you can see in the right-hand portion of the right graph, the CCCP at 100 micromolar really knocked down orange fluorescence, whereas the thiostreptone, on the other hand, actually increased orange fluorescence. And really, so what this means is it's slightly hyperpolarized mitochondria. We don't quite know what this means yet, but we do know that tumor agents, tumor cells rather, have a very narrow spectrum of in terms of the redox state and their ability to survive. So this may be one mechanism which thiostreptone is inducing cell death in mesothelioma cells. Second assay we want to look at is whether or not thiostrepton caused apoptosis. In the nuclear ID chromatin condensation assay is an ideal assay, as Wayne pointed out, to look for the induction of apoptosis. Again, we used the EL406 to seed our cells, as you can see in the kind of process on the left. We then use it to wash our cells after the addition of drug, as well as add our reagent. So here what we're looking at is a true wash as well as a reagent addition, followed by a read with the microplate reader. Again, we use the peri-pump as well as the washer manifold and the peri-pump dispenser tubes, as you can see outlined in the circles and square on the right. Okay, in regards to data, the graph depicting on the left is a known inducer of apoptosis. As you can see, staurosporine certainly induces apoptosis in a concentration-dependent fashion. And really, what we wanted to look for in the right-hand side is we had 4 different mesothelioma cell lines. We really wanted to find out whether or not thiostrepton could induce apoptosis. The bar graphs in red are actually staurosporine with these 4 different mesothelioma cell lines. As you can see, on the left is untreated and the right is treated. In all 4 cell lines, staurosporine certainly cause an increase in fluorescence, indicating there's an increase in apoptosis. Unfortunately, thiostrepton in all 4 cases did not seem to induce the same induction. So our interpretation of this, which although it's negative results, is that thiostrepton is not an inducer of apoptosis, at least in mesothelioma cells. Lyso ID red cytotoxicity assay. Wayne kind of alluded to this as part of the battery of celestial kits that deal with lysosomes and autophagosomes. The Lyso ID red cytotoxicity assay is really an assay that looks for an increase in lysosomal content. One good example of that in terms of drug-induced increase in lysosomal content would be verapamil. And as you can see in the figure in the lower right, with the presence of verapamil, you see a large degree of red staining of the lysosomes. It's really what we wanted to find out was whether or not thiostrepton itself would induce a similar phenomena. Again, the assay procedure on the right, as you can see, there's a series of different processes that we use the EL406. Again, we used it to seed cells. There's two actually wash steps, as well as the addition of reagent. So we've got really 3 steps in which we use the EL406 to perform all the liquid handling tasks of this assay. And again, it's the peri-pump as well as the washer manifold utilized in these assays. This slide's a little busy, but the data on the left are indicators with H-meso cells of known inducers of lysosomal content. The upper one is chloroquine, and as you can see, the red graph really is an indication of lysosomal content, while the blue graph is actually a Hochst dye, which really what we're looking at there is cytotoxicity. So an increase in the lysosomal content with independent of any changes in DNA content from the Hochst dye would be indicative of increase in lysosomal content. And as you can see, both verapamil as well as chloroquine, using the 406, we can certainly get good response in terms of lysosomes without having a cytotoxic event in the cells. Now, the busy figure bar graphs on the right are basically different mesothelioma cell lines, as well as the bottom graph is actually a non-tumor cell line, a mesothelial cell. And as you can see on the verapamil signals on the right is in all cell lines we get an increase in lysosomal content with addition of verapamil. But with thiostreptone, what we really don't see is any increase in lysosomal content, with the exception of the normal cells, which is actually the lower left graph, the LP9 cells. And interestingly enough, increase in lysosomal content has been described as one of the means for cells to accommodate or defend itself against attack by toxic agents, in which case thiostreptone will be considered a toxic agent. And this kind of correlates well with the fact that the LP9 cells are much more resistant to thiostreptone than the tumor cell lines are. And this may be one of the means by which it is certainly less affected by the drug, in that it has maintained a means to defend itself, whereas the tumor cells have lost this means to defend themselves against drug agents. One of the last things I want to talk about is the cyto-ID or autophagy detection. Really, we didn't really work a lot with mesothelioma cells here, but it's actually a work in progress. But as you can see, as Wayne pointed out, there certainly is a means to look for autophagy using these celestial dyes. And again, what we want to do is automate this task. The automation is a little more complicated depending on how you want to induce autophagy. The kind of circular diagram on the left describes either autophagy induced through drug treatment or autophagy induced through starvation. But in either case, we use the 406 to seed our cells. And if we were going to starve them using basically lack of amino acids, we would then use the washer manifold to wash away any of the media, as well as add back a amino acid-free buffer, followed by the addition of our cyto-ID dye, again, and then wash away the dye and read it. On the drug-induced section, where we really didn't need to wash away the amino acids, we have one less step. Really, what we're using is just a peripump to add the drug, followed by the addition of the dye. So you can see that there's less automation steps in terms of drug-induced autophagy in regards to the 406s compared to starvation. But needless to say, in either methodology, we can certainly use the 406 to automate the tasks. And really, what I want to show here is we get really good results either using nutrition/starvation in regards to the induction of autophagy, or if we wanted to use a drug to induce autophagy. And in either case, what we see is an increase in fluorescence as a result of the treatment of cells. And really, what we want to do next is to look at thiostreptone and see if it induces autophagy. We know it doesn't really do much in terms of lysosomal content, but it would be interesting to see if it does something with autophagy. To kind of summarize, I think I've pointed out, and as has Wayne, that the celestial assays are really rapid, sensitive, and specific. But more importantly, in regards to microplate instrumentation, is they're really compatible with high-throughput microplate-based screening workflows. And the 406 is an ideal device to provide many of the fluid handling steps of these dyes, as well as the process required for these dyes. And I think at that point we'll try to go towards some questions. And so let's take a look at these questions and see if we can, either Wayne or I can answer them.
Yeah, if Wayne could answer first, that would be great. We've actually assigned him a few.
Okay.
Wayne, if you can go over to the Q&A tab.
Okay.
See some questions assigned to you.
And should all right. I have a question from Ian Townley at Xavier University of Louisiana. I can't read the rest of it, sorry. Xavier University, though. And I think he says, what is a decrease in the control due to the dye? And I presume that he's referring to the MitoID membrane potential cytotoxicity kit. If you look at the green line, you can see there's a slight decrease in the signal with time. And actually, that's very simple to explain. Basically, that's the cells being at room temperature during the analysis on the instrumentation. So we take the cells out of the 37-degree incubator and work with them at room temperature, and that room temperature does cause a very slight decrease in the fluorescent signal due to a slight decrease in the energetics of the mitochondria. Next question is from Anhoy Besekoya from IDAA. I need dye for cell death cells. Are there any dyes? Again, I think a number of the celestial dyes that we're discussing are appropriate for analysis of cell death. The nuclear ID chromatin condensation assay is very good for measuring apoptosis, as is our nuclear ID our cyto-ID autophagy dye can look at autophagic cell death. We also have a GFP-certified apoptosis necrosis kit, which simultaneously analyzes both apoptosis and necrosis in the cells.
Yeah, I guess I can answer one too. This one's from Nina Bianda. She asked whether or not if I could comment on how washing affects the concentration of cells, and how does one adjust the flow in regards to cells and maintaining cells. The kind of end game of washing with cells and using an automated system certainly is to maintain the cells in the plate. There's a number of things you can do in regards to the washer and the washer settings to prevent losses of cells. What we found in regards to loosely adherent cells is it's not necessarily the aspiration end of things of the washer that removes cells. It seems to be that the dispense end of things is more prone to perturbing cell monolayers. So there's a number of things you can do. Certainly with the cell wash capable washer such as the 406, you can use the lowest flow rate possible. Kind of a standard ELISA washer really doesn't have these low flow rates, but like I said, the low flow rates are certainly will reduce the amount of cell loss. Other things you can do is have a high residual. If you're doing a multi-cycle wash, is not to try to remove all of the fluid every time, but to leave a residual, say 40 or 50 microliters, and maybe on the last aspiration remove more fluid. That residual serves as kind of a buffer to the kind of trauma of fluid flowing into the wells. And the last thing you can do is, at least with the BioTek washers, you can offset the dispense tubes to one side. The dispense tubes on the washer manifolds are canted at 20 degrees. So by moving them over to one edge of the well, you can actually disperse a lot of the energy into the side of the well rather than into the cells. So those are three things that you can do to kind of minimize losses of cells in a cell wash assay. Now, these aren't necessarily things you would want to do in an ELISA assay, but if you have an antibody that's only loosely attached, or your antibody binding reaction in the ELISA itself is relatively weak, then you can use these kind of same functions, if you will, in an ELISA to prevent loss of ELISA signal as well. Another question is asked, is this approach more effective than conventional wash and spin down of cells in a tube placed in the centrifuge? Well, it's not any more effective in terms of the wash procedure per se, but what it does allow you to do is to wash 96 different experimental conditions at the same time. So really what you're looking at is in terms of automation, is the function of sample number and time. I think that's one of the biggest advantages of microplates in general. But being able to do it in 96 tubes at the same time, although very small volumes.
Okay, another question from IDAA, Anhoy. Does BioTek have a mitochondrial dye like a mitotracker for live cells? I need a dye for stained mitochondria from live cells. First thing, Enzo is the dye company. BioTek is the instrumentation company. We collaborate and work together in this area. Specifically, I think what you're asking here is, in addition to mitochondrial membrane potential, can we measure mitochondrial mass with another dye? And we have developed a superior dye for that called MitoID red dye. It measures mitochondrial mass in live cells. It will also measure mitochondria in fixed and permeabilized cells. So you can fix and permeabilize your cells and then stain the mitochondria directly in that context.
I got one here. Is it possible to carry out these assays in a PowerWave XS2 model? The answer is no. The PowerWave XS2 reader is an absorbance-only reader. All these assays are actually fluorescence. So you really would need a fluorescence reader, such as one of the Synergy readers, or an FLX800 reader to measure these assays.
Okay, I have a question from Jessica Bowman, Virginia Polytech. I work with malaria parasite, Plasmodium falciparum. Sorry for jumbling that. Do you have a probe that would target the apicoplast, a plastid-like organelle in the parasite? Would this assay be able to determine toxicity of the drug to the parasite within red blood cells? Generally, the ENZO assays have been benchmarked on eukaryotic cells and not small organisms. You may encounter a dye penetration issue with them, but we welcome the opportunity to let you try. So if Jessica, if you're particularly interested in giving that experiment a try, we can probably provide you a little bit of dye after some discussion to figure out which one might be best for your particular organelle. I suspect it'll be something like a lysosomal dye that you need in that context. Okay, I think we have one last question here that I can probably address as Lissandro from MPI. What is the selectivity of the cyto-ID autophagy kit in other cells like dopaminergic neurons, which also have vesicles containing organelles that also accumulate cationic dyes? First thing to say is that cyto-ID does not really localize the same as conventional cationic dyes. There's a little bit more into the mechanism than that. There are a number of cationic dyes that we have looked at that do rather non-specifically stain various vesicles. That being said, we've tested cyto-IG on a wide range of cells, ranging from primary hepatocytes to a lot of blood cells and so forth. I do not currently have any data on these neurons, so that's one we would have to look into. But in general, we are not detecting heterophagic vacuoles. Those are endosome-derived vacuoles with our autophagy dye. We have a long-term tracer dye that actually works in detecting those kinds of organelles.
Okay, thank you both, Wayne and Paul. We're at the top of the hour here, a little bit over our time. Thank you very much for joining us today, and thanks to Paul and Wayne for their presentation, taking time to answer some questions. We'll attempt to answer some of these remaining questions via email, so if your question didn't get answered, expect a response from us relatively soon. Be sure to check out our website and blog, like us on Facebook, and then follow us on Twitter to hear more about biotech webinars or learn more about our products and how they can help you with your research. Have a great day, and thanks again for joining us today.